Great. So actually, I'm a newbie to the microbiome area. I find it to be extraordinarily interesting, and obviously it's very, very important. But like Howard said, I've pretty much focused on antibiotic R&D for the past 25 years. So this is something new for me, and I'm really enjoying it tremendously. And I want to thank Joanne for inviting me to speak, and everybody for coming here. And I'd also just like to say our labs are just downstairs in this building. So if anybody wants to come by after the meeting today and take a look, we'd be happy to have you do that. Okay, as Keith mentioned this morning, the ATCC Center for Translational Microbiology was formed in a partnership between the Institute for Life Sciences Entrepreneurship that Keith heads and the American Type Culture Collection. And our work focuses on clinical and diagnostic microbiology. We're very much interested, for example, in the commensal flora as a reservoir of antibiotic resistance. We're working in the bioinformatics area, so we have a group of bioinformaticists, software coders, and data scientists that are working to create a next generation automated annotation system for doing genomics analysis. Ourselves, we're doing microbiome and metagenome research, and I'm going to talk about shortly a reference standard reagent that we're preparing that we hope will be of use to the community and that the ATCC will subsequently provide to the research community for this purpose. And we've also working to build academic and industrial collaborations in the translational microbiology area. So there are a lot of different biases that impact on microbiome research, and I'm sure you're all aware with the plethora of publications. I mean, since next generation sequencing, the microbiome field has exploded, and there are literally thousands of publications in that area. And yet, if you look at those publications, there's really been a minimal use of controls, and there's high variability in the studies. Even within the same labs with the similar samples, there's quite a bit of variability. And we want to try to help to address that so we really can ferret out those very fine differences in the microbiome between health and disease that impact on interpretation of the findings. These can include bacterial cell wall composition. So for example, gram-negatives may be easier to extract DNA from than gram-positives, or acid-fast organisms or spore formers. Genome size and supercoiling can impact on analysis. The GC base composition. There are inhibitory substances from biological samples that can interfere with amplification. And if you're not able to extract the DNA, or you're not able to amplify the DNA to do the NGS, then it's as if the microorganisms were not present there. And currently, there's very limited ways to tell whether your protocol that you're using is actually successful in getting that, or if biases are being introduced. So, key steps for quality control of microbiome research include a variety of different aspects impacting on sample sequencing, sample processing. And it's a little hard for me to see the screen here, so I'm just doing this from memory because of the sun. The sequencing analysis and also the data analysis impacts on a variety of different processes that can impact on the findings. It's important to implement a QC protocol and procedure into your work to address this. And for that, an appropriate reagent is necessary, whether it's an internal standard, external standard, but a variety of different opportunities are present there to address these potential types of biases and problems. Okay. So we have deep expertise in this field. It's unfortunate that Raul Cannon couldn't be here today. He's our director of microbiome research, and it would have been a much better talk given by him because that's really his expertise as opposed to antibacterial discovery like mine. But we really are working towards spiking controls, mock communities, so where we have collections of different bacteria that are representative of microbiomes, diagnostic internal controls for quantitative PCR, synthetic matrices to look at, for example, inhibition of amplification by bile acids and other pigments that are present in fecal samples, and sample collection tools. So if you're familiar with the microbiome literature, even something as simple as whether a cryopreservative is included into the biological samples that are frozen can impact on the outcome of the findings. Okay. So one of the things that we're interested in doing is preparing these reference reagents. They're internal competitive controls. These are for spiking in and looking at these biases. But in addition, they provide an opportunity for normalization of data. They provide an internal reference to compare your findings, your microbiome, your gene quantitation from metagenomic analysis. And at this point, we are working on a variety of different bacterial species. So we're creating a lactobacillus strain for this purpose. We're creating E. coli and many other types of bacteria for this purpose to address those differences between the species that can impact findings. The way that we're going about doing this is we've created a tag, a barcode, okay? And this is a chimera. So this sequence can be detected, and this sequence can also be subtracted out by software, and we use QIIME to do this, okay? But we have on this DNA sequence, this fragment, we have the variable regions of the ribosomal RNA that's used commonly for taxonomy studies in the microbiome. We have a synthetic sequence, and we have the barcode that we're able to monitor. We've created a variety of different formats. The two that we feel are the most important are actually an oligonucleotide that can be used in part for library construction quality control, but also we've made recombinant strains, viable bacteria that have a single insert in the chromosome, so we can address these extraction and amplification biases by spiking this into the samples. And you could see here where we propose that this type of reagent or these recombinant bacterial strains would be useful in the process. We've gone through and we've done validation so we can utilize the most often utilized primers for doing amplification of the variable region. And we're able to detect our tag. We can also look at and differentiate the native variable region from our barcoded insert. And we can use this to do library analysis and also to quantify this and detect this with Agilent Bioanalyzer. Now, this is also intended to be quantitative. Okay, so we've done studies where we actually put different numbers of molecules of the oligomer into the studies, extract, amplify, and sequence. And you can see here from this slide the recovery of the tag sequences. And this is utilizing, again, the primers for the ribosomal RNA variable region. Okay. So this is our process, for data analysis and for our validation. We denoise, clean up, and demultiplex. We identify the operational and taxonomic units, and then we normalize resulting data, and we can subtract out this signal so it doesn't skew the microbiome, because you can imagine compared to a single species in a microbiome that may be very important but may be in low incidence, we're actually spiking in a lot of signal, whether it's E. coli or lactobacillus or so forth. So you can see here where in the next to the right, the left figure, the next to the right column is actually the change there is due to our tag. And then you can look at these microbiome analyses on the right side where we've actually subtracted that out using the software. So, and you get a much better quantification and ability to compare things with this type of internal standard. Okay. So I wanted to show you one example of where this may be very important. So we're collaborating with many people. We're collaborating with physicians, we're collaborating with other universities, we're collaborating with commercial farms. You know, we're very interested, obviously, in antibiotic resistance, and while the US government and the Europeans have curtailed the use of antibiotic growth promoters, you know, in the developing world, there's still going to be continued and expanding use. In fact, the Pew Research Foundation study shows by 2050 the explosion of food animal agriculture with the increasing population in the world and like a wild west of antibiotic growth promoter use. So this becomes really important because of transglobalization. We'll continue to have significant antibiotic resistance, even if growth promoters aren't used here. And when you look at this type of analysis, so this is a pig fecal microbiome analysis without using the tag sequences and normalization. And you don't see a lot of diversity there, or you don't see a differentiation between different groups of the animals with this type of analysis without using a reference standard. But after this, you know, where we're able to standardize this and normalize the data, we can see marked differences. And in fact, we are able to differentiate animals that have received antibiotic growth promoters from those that have received an organic diet that enhances weight gain just as much by having a certain type of food additive and the differentiation there in the microbiome. And in fact, I'm sure you're all very interested, you know, with the lactobacillus. This correlates with an increase in the lactobacillus species and numbers in the pig microbiome here in the animals that did not receive the antibiotic. So we've designed 10 different barcoded tags. We can actually utilize multiple bacteria with this, and we can seed in all of those and look at these biases. We have it, you know, based on the different biases: cell wall type, genome size, GC base composition, aerobic, anaerobic, spore formers, non-spore formers, so we can address all of these potential issues. As Keith mentioned this morning, our colleagues at the ATCC are working with NIST and sponsoring work in this area, and we hope that we'll be able to prove this out and be able to provide these reagents to the community to really enhance the work and further look at those fine differences where some bacterial species may be very, very important, but the variability really precludes making a definitive discovery. I think some of the other things that this is useful for as well, one of the things we're envisioning is actually metagenomic modeling, where we can take these bacterial species and we can feed them to animals, okay, in a controlled environment, and we can differentiate what happens to the tag. So we can follow those bacterial species through the microbiome, okay, as we're you know, doing model manipulation to try to understand, or even 2 or 3. So we could feed an E. coli that has a tag, a lactobacillus that has a tag, and then we can monitor what occurs with those. All right, so I would just like to thank you for your attention. Be happy to take some questions later on. All right, great. Sure, Howard.
Monitor the genetic drift of the organism? I mean, have you thought about that?
Yeah, we're thinking about that, and we're actually thinking about putting this type of tag into mutator strains. You know, one of the most common things that occurs and is very problematic with antibiotic use in animals, as it occurred in the past, right? Tetracyclines. I think 70% of all the tetracyclines are used, and tetracyclines, you know, select for mutators.
Yeah.
And you get cross resistance against different classes because of that. So it's really, really problematic. So yeah, we're thinking towards actually looking at genetic.
Yeah, and I wonder, were you at the NIST meeting that was at the end of August?
No, I wasn't,
Okay,
unfortunately. I was
because
registered,
so
but I wasn't able to attend.
there was a meeting at the National Institute of Standards and Technology about these very issues.
Sure.
At the end of August, and I didn't attend, but it was very well attended from people all around the country. So
Right.
these are important because, like I said, what is the core standard organism for standard sequence and how much genetic drift can be tolerated before it becomes a different organism.
Exactly.
So I think that's
No, I agree with that. We're
important
doing a
issues.
variety of things too. We're making a version where we have all of the primer sites for all of the variable regions in one product. So whichever, one insert. So whichever different researchers prefer to use, or they can even look at.
Yeah. And just out of curiosity, are you working with any of the big food companies like Danone on this idea?
We're not working with them. This is for the ATCC.
This is just for the
ATCC
ATCC.
would provide this to the community.
Okay.
Actually, we're a nonprofit also.
Right.
This is for the benefit of all
Sure.
microbiome researchers.
Yeah. No, I think it's important because the field is exploding and there has to be some control and standardization of it. So.
Exactly. Well, I mean, just this past year, we were working on a grant proposal, and, I'm looking at the literature on the Fusilibacterium, and I read 6 papers. Well, 2 of them say it's important in the levels, and the other 4 say they don't see anything.
Yeah.
So this sort of reagent can help.
That brings up the whole issue of mouse studies, because there are some people that feel mouse studies are inherently non-reproducible because of the biome differences in the different colonies.
Right. Well, I think that's important. I think another couple issues. Oh, I'm sorry. What do you want me to do? Oh, I'm sorry. Yeah, a couple other issues. So, yeah, about a year or so ago, I left GSK. I was working there, and the microbiome research there was done in mice, and they really stopped the work because they just didn't find anything in, metabolic diseases and diabetes based on looking at the mouse microbiome. Our particular predilection is to look at the pig model.
Yeah.
And this, we're collaborating with a research veterinarian at Cal Poly out in San Luis Obispo. So,
Are they using miniature swine?
well, he actually is using, and this work was done in the red wattle, but we're moving towards actually looking at the Göttingen mini pig also. Different breeds are useful for different purposes.
Sure.
So the Iberian, you can look at fatty liver disease. This particular species, our interest was in looking at our tags for analysis and also the growth promoter area and trying to help to get rid of antibiotics or understand antibiotics, because we're very interested, like I said, the commensals as a source of resistance genes, right? So it kind of makes sense to look at that. But yeah, we're looking at the mini pig
Yeah.
too.
Unfortunately, I don't think that would work at NIH because there's enough pork in Washington now.
I agree. So.
Yeah, I'd like to just add a couple of comments beyond what John has already said, and thanks for the very nice presentation. We are in the process of setting up a microbiome network to help us, to guide us in the creation of future tools, and as I said earlier, to identify beta sites that could test some of the reagents and tools that we're developing, and also to create a forum similar to this, perhaps, to actually talk about issues in microbiome research that go beyond just the findings, the directed scientific content findings of studies. So we're not there yet, but hopefully when we get there, you'll take a look at our site and maybe consider to join. The other thing that I'd like to add is that we're very much interested in collaborating with groups. We're interested in developing a new model for microbiome studies, particularly gut microbiome studies that would be more relevant to man. And we're very interested in collaborations and sponsored research and other types of grants where we could really add our expertise and our tools and reagents and interest in the microbiome space with other companies and academics in the community. So if you have any interest, follow up with John, and thank you.
Thanks, Keith.
I'll just mention that I started the microbiome listserv at NIH, and so we could certainly use that as a venue to propose ideas and see if there's, at least within the NIH and FDA intramural communities, there would be an interest in working with you. So we'd be happy to do that.
Well, I think one thing too, like Keith was pointing out this morning, we would love to find collaborators to help us test these reagents, okay, to confirm their validity. So, all right, well, thanks very much.
Okay, thank you.